Okay, so the next part for today is what? Last time we did the test for protein, eh? So, but what chemical do you use for testing for protein? Burette, eh? Then what is the color for burette? Blue color, eh? All together?
Yes.
Then if the protein is present, what are the changes that occur? Purple, eh? Purple, eh? I know that some minor colors, but the major color is purple, eh? Okay? Then if it's absent, what happens? If it's absent, what happens?
Do not change.
Don't change, right? So do not try to don't change. So the solution remains what? Blue, right? So it says the solution remains what? Blue. Okay? Then we can give an example of food that contains protein. A protein, fish, what else? Fish, what else? Fish, What? Egg, okay? What else? These are okay, good. These are okay. Then, what disease is caused due to lack of protein in the body? Marasmus, eh? Kwa sugar is for what? For carbohydrates, eh?
Carbohydrates.
So marasmus, eh? Marasmus, okay, good. Then, what about starch? Which chemical do you use to? Okay, before we go to starch, the method in terms of protein. What are the methods? What are you supposed to do? For example, like if this sample is given and asked you to test for protein, what can you do? If the sample is like this one is given and asked you to test for protein, what are you supposed to do? Any idea? Yes. So you don't say add Dico solution or whatever solution. You need to come up with a method, right? So you need to write your method clear, right? Step by step. We do this from there, we do this all together. So now we can try to explain step by step. Yes, ma'am. We don't add. For example, when you get this test, this is a test tube, right? This test tube example is empty. So when you say add, which means there's something already. You are just adding on, right? So now the test tube that you are getting, is it empty or there's something already? Empty. So why are you saying you add? So put. So solution P. So if the solution P is given, use P altogether. So put 2 centimeter cubic of solution P in a what? In a clear and dry test tube. Why? Then from there, how do you do? You add what? So you put 2 centimeter cubic of burette reagent. That's the method, right? Then now observation. If it's present, what happens? If it's present, it tells what paper, right? If it's absent, remains brown. Blue, right? Good. Okay, what about for starch? For starch, what happens for starch? For starch, method for starch? And remember I said that for protein, the sample is supposed to be what? In liquid form, right? So if it's in solid or powdered form, make sure you mix it with water, then you filtrate. All together you get the liquid by filtration, right? So now we go to what? To starch. Who can give an example of food that contains starch? Food that contains starch, Potatoes, shima, right? So these mostly they come from plant, all together from plants, right? Good. Then, what chemical do you use for testing for starch? Iodine. What color is iodine? And if it's present, what happens? It is blue black, eh? Good. So now today we'll go to the next stage, which is test for reducing what? Reducing sugar, eh? So reducing sugar. So now about reducing sugar, we mean a food that contains sugar. Example like Apple Max. What else? Even Coca-Cola, small man. Even a drink. Even this power what? The sugar here. The not sugar. So when you test this one, the sugar will be present. So good. So testing for reducing sugar. So how do you test for reducing sugar? So first we need to know the chemical that are involved in testing for reducing sugar. So altogether, you need to understand the chemistry the chemical. And we call it the Benedict test for reducing sugars. So we call it Benedict test for reducing sugar. So it means the solution or the chemical that is used to test for reducing sugar is Benedict solution. All together. Again, we need to know the color. The color bwanji. So now even ya blue. Tamela point is blue in color. So burette na Benedict solution ni pipara na makala zote. Ni blue. All together. Good. Then from here, you need to understand now the method, the procedure. So they're saying that if you want to test for reducing sugar, this test requires the sample to be in solution form. All together? So it means the sample need to be in solution form. In this form, all together, like liquid. So even if the solid is given, one percent of it solid, whatever, you need to try by all means to convert it into what? Into liquid by mixing it with distilled water. All together, you grind it. You know grinding, Once you grind them, then mix it with distilled water. Then you do what? You filtrate and get the solution. If the sample is in solid form, it will first need to be ground or crushed, cut into very small pieces. Example, if a meat is given or beef, so pieces, then you put it where in distilled water together. Then now you get the liquid all together. So you cut very small pieces and to be shaken with a distilled water for extraction of reducing sugars. If they are present, filter and then proceed with the flowing. So now, how to test for reducing sugar? The method. So number one, you need to put 2 centimeter cubic of what? This is a method now. You put 2 centimeter cubic of sample solution in a clean and dry test tube. So you get a test tube. Then you get 2 centimeter cubic. You can get 2 or 3, whatever volume you want to get, but be specific. So you can say, get 2 centimeter cubic of sample solution P and put it in a clean and dry test tube. All together. So you get 2 centimeter cubic of solution P and put it in a clean and dry test tube. Okay, just say put 2 centimeter cubic of sample solution into clean and dry test tube all together. Then from there, add 2 centimeter cubic. In short, you put equal volume, but in my exam you don't write put equal volume. So now solution mafaka 3, you also say put 3 all together, but the fact that you're supposed to use equal volume. So you say add 2 centimeter cubic of Benedict solution to the sample and shake. So after adding the Benedict solution into shake all together, you shake all together. Then after shaking now, you gently heat the mixture using water bath. So you can say maybe for 3 minutes or 4 minutes, whatever time all together. So which means for reducing sugar, you need to heat this one, but you heat it gently all together. So I'm saying that number one, the solution or the sample need to be in solution form all together. If it's not in solution form, you crush it or ground it all together. Then you mix it or cut it into small pieces. Then you mix it with a distilled water. Then you shake it like this side. Then you filtrate the liquid and test the liquid for reducing sugar. So now if you have a liquid now like this, one, what are the procedures? So what do? You put 2 centimeter cubic of sample solution in, maybe it's P, okay, in a clean and dry test tube, right? Then you add 2 centimeter cubic of Benedict solution and shake all together. Then you heat the mixture gently for 3 minutes. All together. You heat the mixture for 3 minutes. All together. So that's the method. Then now after doing the method, you now go for observation. You observe what happens. So who can try to explain the method before I go to the next part? Who can try to explain the method first before I go to the next part? Who can try? Let's try. The method is, it's very simple. It's just like you are just chatting, right? You say, oh sir, for example, if the sample solution, most this time around they give what you call, they give in liquid form, right? But in case they what if they give you onion and tell you to test it for reducing sugar, what you can do? Onion, onion, eh?
Yes.
Onion with sugar, onion, eh? Oh, I don't know. So if they give you onion, what percent onion is that? Onion. What can you do? You cut it into small pieces. Or crush it, right?
Yes.
You crush it into smaller pieces. Why crushing it?
For us to be able to taste the reducing sugar, we have to put the given sample into a solution form.
So it's supposed to be in a solution form altogether.
Yeah.
So you say because for testing for reducing sugar, it needs a sample to be in solution form. Therefore, you need to crush it, then mix it with what? Mix it with the distilled water altogether, so that now you can get the liquid. Then you mix it with water, then you shake papa. Then from there you filtrate, you extract, or if you get the liquid altogether. Then now you after getting the liquid, you proceed with the following procedures. Right? Number one, what do? You put 2 centimeter cubic of solution K into a clean and dry test tube. Then you add 2 centimeter cubic of Benedict solution and shake, right? Yeah. Full stop. Then again you say you heat the mixture gently for 3 minutes. If you want, you can say 4 minutes or 2 minutes, whatever. Okay. All together? Yes. So method is clear? Yeah. Then now, observation. What you are able to see, what you observe. So if the solution remains blue, which means the reducing sugar is absent. So if the solution remains blue, the reducing sugar is absent. But if it turns green, yellow, orange, brick red, which means the reducing sugar is present. So sometimes you can able to see all those colors. So example, green meaning at turimo, yellow yambiriko, orange yalimu yambiri at least, brick red ni shizoda there is a lot of sugar. That's why in the lab we are not supposed to test everything. You know that even a urine example even here we can test we can know that this person has sugar disease or a sugar disease. We can just tell you to urinate in this carbotrome, after carbotrome, we'll get the urine. Then we're going to test this urine for reducing sugar. You know what we can do? We can just get your urine, then we put it where? In a test tube. Then we add equal volume of Benedict solution altogether. Then you do what? We shake. Then you heat for 3 minutes. After heating, if the color change from blue maybe to green or to yellow or orange or brick red, you know that this person has sugar disease. That's why mlaba mali sakuja sometimes ungadabwe mitundo zinga tukapita kwa hospital from a patient who is what aina sugar. Then manage sugar disease altogether. Then what will happen in life from carbotrome? They can add the chemical that you will reduce the odor, the smell, right? Altogether. Then now they put in the lab. Then uzaona at couple max. We never ask at couple max ama kazi ni mitundo za mundu. So that's why in lab we don't need to eat, don't taste. Altogether. Don't eat. Altogether. Yeah. Yes. So using sugar, we can test even urine, drinking, whatever, drinking a sugar like Fanta, Coca-Cola, Apple Max, all those we can able to test altogether. Sometimes they can use what you call sugar. They can just they can add water to make a solution and ask you to test for reducing sugar. So if the color changes from greeny, brick red, orange, whatever color, just know that the reducing sugar is present. Right? But you only write the final color. It can be greeny. The last color that you yourself have observed or seen. Don't write greeny, yellow, whatever. You just need to write the final color. The final color. All together?
Yes.
You just need to write the final color. The final color. All together?
Yes.
Good. Okay, proceed. So now, is it clear on reducing sugar? So number, don't forget the chemical that is used to test for reducing sugar. Because next time as I come, as I said, you just find my apparatus. Then a negotiation paper. Then you'll tell you, okay, the first step, write the method. How to test for maybe starch. Right? So you just write, do this. Conclusion, you test, you write your conclusion of observation. Together. So even a final exam practical, when you go for orientation and final exam, that's what they do. They just give a question paper there. They'll just say, test for starch, method, you write your own method. All together? Yes. So this one is clear. So now we go for non-reducing sugar. Or before that, they're saying that only state the final color observed and not all the colors mentioned in the table. All together, as I said, the extent of the color change indicates the quantity of reducing sugars present. Green and yellow colors indicate the little or traces small amount of reducing sugars are present. Orange indicates the reducing sugars are present. And brick red indicates high concentration of reducing sugars present. So those colors, they just show, okay, there's some traces, okay, there's reducing sugar. Now here there's excess reducing sugar. This is what together? So in short, on this one, conclusion is that you only write the final color. Is it clear?
Yeah.
Then we go to the Benedict test for non-reducing sugar. Kuziba kuti umumude sugar. Sometimes you cannot even know that there's sugar or no sugar. You have together, like onion. Onion in a sugar, maybe sugar onion. In short, some of you don't know that there's sugar in onion. So now they give you onion test for reducing sugar or test for non-reducing sugar. So what you need to do, to test for non-reducing sugar, first you need to carry out the test for reducing sugar. Right? Then you, after observation, that there is no sugar, you now proceed with the non-reducing sugar. So which means the first stage you need to do a test for reducing sugar. So I'm saying that if one test for non-reducing sugar, first you need to do what?
To test for reducing sugar.
First you do what? First you test for what?
For reducing sugar.
First, you carry out a test for reducing sugar by using the previous method. What I'm from explaining altogether.
Yeah.
So, pay attention. The Benedict test for non-reducing sugars. First, carry out the Benedict test for reducing sugars. If the color of the solution remains blue, proceed with the next step. So, which means first you need to test for reducing sugar. And if it's present, if the color remains blue, then now you proceed. But kapezekati ya chinja, you cannot proceed together. It was what the sugar here. It was what the sugar here. Clear? So now, how do you proceed? You proceed by putting another, so it means the solution that you have used to test for reducing sugar to confirm if reducing sugar is present or absent, you get the new solution. Right? Yes. No, if it's not present. After testing for reducing sugar, if it's present, if it's not present, you proceed. So, yes. First, you test for what?
For reducing sugar.
Some cafe that it's absent.
You
proceed.
Yeah.
Hey, Kaish maziwa timlibe, so amen. So now I want to confirm if reducing sugar is present. Eh? Another point, eh? Yes. Out together.
Yeah.
You're confused again. You're confused. Right. So now, I said, first you carry out the test for what? Then mkapeza ili absent. Right. Then, example, testing, maziwa talk here is not present. What do? Do you use the same liquid or maybe you get the new one? The new one, eh?
Yes.
Good. So you place another 2 centimeter cubic. Continue manage. After maziwa talk is absent. Eh? You place another 2 centimeter cubic of sample solution into clean and dry test tube. All together. Then you add 1 centimeter cubic of hydrochloric acid. All together. Then you heat in water bath for 3 minutes and cool. So when you get a new solution, you put 2 centimeter cubic of the sample solution into clean and dry test tube. Then you add what? Hydrochloric acid. Hydrochloric acid. Together?
Yes.
Then from there you heat maybe for 2 minutes or 3 minutes, right? Using water bath. Together. Then after adding this acid, next stage you add sodium hydroxide. Together, which is sodium hydrocarbonate or sodium hydroxide solution to the mixture. Little at a time until fizz stops. Okay, there's acid there, remember? Acid. Then now you start adding now a base. Okay, sodium hydroxide or sodium hydrocarbonate is a base. It neutralizes acid. It neutralizes acid. So mkabofaka acid because kuzaka mafizi, it means acid is being neutralized. Okay, first you add acid, then you heat for 3 minutes. Then now you start adding what? And okay, you allow it to cool first. After adding acid, you allow it to cool for some time. Then now you add what sodium hydroxide, which is a base. Then little by little, drop by drop, bit by bit together. Then you continue adding until the fizz stops completely. All together. Then after now add the sodium hydroxide and the fizz stops, you now put equal volume. Example, which is 2 centimeter cubic of Benedict solution. Solution. All together. Then now, after adding the Benedict solution, what do? You heat the mixer gently using water bath, and then you observe what will happen. So this is the method. So I said, number 1, what do? You test for reducing sugar, and if the solution remains blue, you now continue. You put 2 centimeter cubic of sample solution into clean and dry test tube. Then you add half, which is 1 centimeter cubic of what? Hydrochloric acid. Right? Then from there, what do? You heat the mixture gently using water bath for 3 minutes, right? And allow it to cool. Right? Then you add sodium hydrogen carbonate or sodium hydroxide. All together. Then little by little together until the fizz or fizzing stops. Then you add 2 centimeter cubic of what? Benedict solution. Then you heat the mix out together. Then from there you observe what will happen. Makana procedure akakaribu difficult the procedure. So be very careful. Very careful. So hapa now tutazama pamoja. Number 1, I said you do what? You first carry out test for reducing what? Sugar. So what's the procedure for testing for reducing sugar? Put 2 centimeter cubic of sample solution in a clean and dry test tube. Then you add 2 centimeter cubic of what? Benedict solution, eh? Then you heat the mixture gently for 3 minutes using water bath. Then if the solution remains blue, then now you proceed. The testing for non-reducing sugar. So now you get another solution, another 2 centimeter cubic of the sample solution, put it into a clean and dry test tube. Then you add 1 centimeter cubic of what? Hydrochloric acid altogether. Then you heat the mixture altogether for 3 minutes altogether. Then after heating, what do? You allow it to cool. Then you do what? You add sodium hydrogen carbonate or sodium hydroxide solution to the mixture, little by little, until the fizz stops. Altogether would disappear. Then now from there, what do? You add the Benedict solution, which is 2 centimeter cubic. Then now you observe. Altogether, step by step, you observe. Fine. So after observing now, let us see what will happen in the table. So the possible observation and corresponding conclusion are given in the table below. Solution remains blue. Non-reducing sugar is absent. Solution turns green, yellow, brick red, orange. Non-reducing sugar is present. Have you seen that this one is just same with reducing sugar, but the ketchup you just add acid and the base altogether, and the base. Nishi kwasila mwate explain. Unless if there are some issues here, you can ask me. Question, mommy. It's clear? This is very clear, but it's very tricky somehow, right? But it's like this one should be a song. Each time you sing it, it's just like you're telling a story. So you do this, it's very simple. Or when the exam, or in vernacular you can able to know it, but when you're writing using English. So that's the first stage. First for testing, first for reducing sugar. Mukapeza mulibe, you test for non-reducing sugar. But mukapeza mulimo, you don't test so many. You stop there. But mukapeza mulibe reducing sugar, you proceed. Now how do you proceed? Okay, sasa mzatenga sample inangu, then mzafaka mkatest to clean 2 centimeter cubic. Then mzafaka mu acid, mkafaka mu acid ija. Then mani mzaitinga, then acid ikakulinga ko pangono mfaka mu sodium hydroxide. Sodium hydroxide icha neutralize acid. So that neutralize acid manje paja ngufaka mjeti drop tumono until acid itakuwa until mafizzing ya asiya. Akasiya mafizzing aja, then manje mushtabandi mfaka 2 centimeter cubic of what? Benedict solution again. Then again you mwaitinga futi. Mukaitinga manje paja. You now observe the color zai. Ikachinja brick red, green, whatever, that's what non-reduced sugar is present. And as a change is absent. Is that clear?
Yes.
Good. I know we did on the burette test for protein, we did this one, right? So that don't feel like natural data, eh? So go straight pali testing for lipids, my fats and oils. You know my fats, right? My fats, my fats, right? So we don't want to for fats, what's the difference between a fat and an oil? Mungambe bwana question. What's the difference between a fat and oil?
One is in solid, one is in liquid.
At my fat and oil. Fat and oil. What's the difference between fat and oil?
One is in liquid or one is
Oil is what? Liquid, eh? Fat is solid. All together? What about a tree and a plant? What's the difference between plant and a tree? A question about what's the difference between plant and a tree? How can you explain it?
The difference is the same, sir.
Hey, what's the difference? Difference is the same. Are you sure?
no.
Yes, what's the difference? You know, but baza kulitano mwekunyumba bali. Ango. What's the difference between a plant and tree? Mukamshote mwana ati opanga chongo nayo kwa veki go there. You know, these my kids there. Okay. So you're going to say that to me so much. When you say this is space. This is space, right? When you say. So okay, we'll go to the testing for lipids and oils. We're going to take a to salad. Cooking oil, right? Or sometimes or animal fat. You know animal fat, huh? Animal fats are tested for we call it the emulsion test. Right? We call it the emulsion test. Emulsion test all together. So how to test for testing for lipids? Malipids, right? Malipids same na mafuta, which is the fat and the oils all together. Sometimes they can give you in a groundnut. Not groundnut contain oil. Groundnut that tunakuza tipone groundnut. We forget man. Sometimes even unga wa shaba, you know unga wa shaba? Baenda kushughulikia wakuza ni manda tenga ni manzo. The testing abanje. You take ufaka mtu manzai, mtu manzai, then you get the malipi, then you can have the test. So for 74 dabwa. Ukafuata tamba topa tushaba paja za ta rinso hapa. I'm testing for lipids, right? So think about the method. You know the method all together. You know it's simple. So test for lipids, oils, right? The modern test. So now the procedure: you shake a sample, small sample, or drop of sample solution. Be careful. So we are saying that you shake a small sample or drop of a sample. So it can be this sample, you get take a drop altogether, you get take a drop, or sometimes you can get even to groundnut altogether, small sample of the groundnut. So you shake it with what? You shake it with what? With 2 centimeter cubic of ethanol in the test tube. Yeah, so we get ethanol. Altogether. Yeah, so get ethanol. Ethanol alcohol. So that's why some people, you know, nagashokanya nyama muza mafaka mo bottle mafaka mo vinegar so to me that time to neutralize the fats. Yes, kenda kuja mi chopo kama tumeenda na bottle mtu makakata ka spirit ka isa na mimi trapo mo trapo buka all together, all those things. So you shake small amount or small sample or drop of sample. So this one you can just get it a small sample in powdered form, kapeni kanyama all together, kapeni kanyama, whatever given, small sample, just or even powdered, even liquid form like this, just get a small drops all together. You shake it with what absolute what ether in a test tube all together. So you shake it. Then after adding ether now, you add few drops of studio to manze to the test tube, then observe and record what happened. So I said about what you do, you get small sample or few drops of a sample given, you put it in the test tube, then you add 2 centimeter cubic of what? Ether all together. Then now you add few drops of water, distilled water all together. This is the method is done. Clear? Then now you observe. If the solution remains clear, you can say clear. Nishmazibati. My fats, oils are absent. Kaya mafuta manzi. Just imagine pa oil paja. Muza machitika mafuta muri oil, muri mafuta mafuta manzi. Muza machitika mafuta mina watu makumbi cloud so men. Cloud so men. Shimaziba ti mafati hivi. Ochitumboa wakati chitumboa mwana machitika chitumboa. So if the solutions remains clear, my fats or oils are absent. But if the emulsion formed solution turns cloud, cloud so white so mwana machitika mafuta nguku mukafaka manzi mafuta mapa mamba kwa watu makumbi so men shimaziba okay, this is what now people say ethanol kai maningi sakoti mafuta vya bichezo then mukafaka manzi mali za bapa mpaka na cloud manzi. So people say kuti atiyofaka mo vichani vya ethanol ni hapo bichezo va together then mukatata manzi mali vibura pa mamba manzi za cloud. So if emulsion formed or solution turns cloud, then fats or oil are present. Present. All together? This is the test for lipids. All together? So when a lipid or sample is given, you just get a small sample. If example I give you millimil or maybe groundnut meal with wakaikaitenga, you just put in a test tube. Then what do? You add ethanol. Then you shake. And then you also add water together. And so if the emulsion is formed or clouds, that's not it's what's present. But if it's made clear, it's absent. It is clear. So by the time I repeat sense of this by now, this is very simple now. You have known the process, eh? Please don't give up into the gun. Can I go to the last part? When I conclude, you are going to the last part. Can I go to the last part?
Yeah.
The grease spot test. The grease spot. So this one, you place a drop of sample solution on a filter paper or brown paper. So you get a filter paper or brown paper together. Something like a filter paper in there. Paper tab paper. So not mukadaba example mwenda exam wakusani paper. Tutengane paper hiyo mukadaba na paper nyacha paper. Yule mbelako. Yule mukadaba sawa nipasa paper kuno kule. Acho kaenda mtoje tu mafoni. Sawa nipasa paper kwano ka paper ka. Wale WhatsApp ka paper kitabanya ka paper. Hapo ndio yule mwa exam yule. Nakati ya telefoni kati ya telefoni. Come together. So you place a drop of small sample. So you just get a small sample. Or even like a drop tab, like a drop can do only if you only filter paper or what? Or filter paper or something like they can give you a brown paper. So I mean, good. Then from here, you place a drop of distilled water next to the drop of the sample. So example, this is a drop. Then now you just get commands not together. You get a small sample. You put in a white paper. I mean, on a filter paper or brown paper. Then you put like a drop of distilled water next to the sample together. Then you hold the paper against light. So now chebe wina ngachoka mikiza mwina aka drop afaka drop ndo ka drop. Then atingana manza tofauti manza tungono. Then abachoka panja mchaso kazima so. But afunda uta. Can't how it's a procedure. Kama ngatisonga dabo na mwana mzaza atatenga mzano esi mkai ukapenda question ngomu ngalibo nofika hapo ndasema mzako atenga cheka chani katana aenda pakazo kusaka kazuba kanzi saka pakazo kazuba sima dabo atafunda kufunda bazo sima dabo kufunda so meme but it's normal. So sometimes they can give you maybe source of heat where you can go and place your paper all together. So you place a drop of distilled water next to the drop of the sample. Hold the paper against the light until the drop of water disappears. Then observe and record what happens. So, if observation, sample spots so kaka disappear kwa ni absent. Then kaka forminga if a permanent translucent or kaka oily or grease so nishimazeba ati grease spot is present. Because you know example like chitumbua. Udamu watatu mkagula chitumbua bakafaka paper vile. Bakagula sala jambi vikafuminga vikayuma vikankalira permanent chani vile. If tumbua mangana na chani na chani na sala jambi so amen. But kaka tungono vayuma tatu lazima tasadi ni ngono So mkaona vacha disappear complete just not grease chani spot is absent. But kapezeka vaforming permanent grease spot or oil so maziwa is present. It is hard. It's hard.
Yes. So when you place a drop of water, you're not supposed to place it like that. They're not supposed to mix. Drop of water is supposed to be.
No, it's kind of next. So it means you can even meet. There's no problem. Kai people put it command as well. Ndo up next drop do. Haya. Funga vakumu ya nasome. Good. Question?
So is to go outside.
Yes, you can go outside. Go outside, then you look for the sun. Why you do like this?
Can they agree?
It's part of exam. I mean, they can agree, yes, they agree. You go outside. All together. You do like this, Kasani. Then now, disappears that okay, it's absent. But if the permanent, translucent, or grease or oil is formed, that's what is present. You go and write now present. All together, family. Very clear? So all these things that I'm telling you, my exam will go with my exam, saving Karamwe. All the methods supposed to know them. So na nenda mrudi makani visa mrudi nenda mrudi visa batabio. In case na pesa mtocha ma practicals. Zafika tabo, what are you doing? Test for starch. That's all starch I use iodine solution. What are the procedures? I know the procedures. So finga tevi chebe mti matabe once finga tevi visa ba. Because mja exam kuli bat nikachenge mbuka chebwanda you need to write. All together. You need to write. So this method. So provided you know the methods. Or maziwa top the iodine there. There's burette here. There's what there. There's ethanol there. You know, ethanol I need to use it on that one. There's what else? Sodium hydroxide was ever talking in test for what? There's hydrochloric acid was ever hey manga label. So it's up to now just to know the method. It's up to you to know the methods. So on the next time I just put the apparatus here. I have them there. I put them. Each person will give I give you more or her the paper question paper. Method wa Muslim direct. Mkaone vile mzaka nema manje. Sasa natoka mama belenga soma belenga. All together. So just prepare next weekend. This weekend. I just come for biology practical. Zapita nafaka kwa apparatus hapa. Papa mshimoshe ponsa. You test testing now one by one. But just naenda kupaja you write the methods. No phone, my phone also kusia kusogolo. You will see if you're able to remember the procedures all together. So what's important is knowing the procedures and also what happens. Because even in paper 1 and paper 2, these things they come for an exam all together. I think so last time you saw the questions on paper 1, right? Yeah. Paper 1, yeah. So on paper 1, my question, example, what will happen if the starch is present, if it's absent? You know, you need to know these things. Even in paper 2, theory, just imagine theory. They can able to ask you a question. Like last year, question last year came. You saw the question about onion. So I'm testing for onion. So you need to know all the procedures. Unless if there's a question, you can ask him. If no question, you can end here for today for biology. Any question? No question. Human question. Another question. You have come late. Question. Question there. Any question? No question. So if no question, I think so.